The biochemical tests. Okay, so the purpose, doesn't change, and it just like with the stains, all the purposes are listed on the PowerPoint, right? So be sure you're using the PowerPoint for these, right? That the purpose of this is to help you with quizzes and the practical, and if you don't use the PowerPoint, then you're not learning the material for those. All right, so the biochemical test, the principle is a little different than the stains, okay? The directions for one test may not really pertain to another test, right? Because they're not all, they're biochemical tests, but sometimes, you're just testing for a particular enzyme, and if there is an enzyme listed in the PowerPoint, I expect you to know that that's the enzyme needed for that particular test. Or, basically you're saying if the bacteria has this enzyme, then they can break down starch. If they have gelatinase, they can break down gelatin. So we call the gelatin test or gelatin tube media, a biochemical test, and that answers that specific question. So, just like with the stains, you're still going to be putting in what you see on a PowerPoint. Use complete sentences. If you don't use complete sentences for your principle, then you lose 1 point out of the 4. So just like before, you get 1 point for the purpose and 4 points for the principle. And you need to put down what indicates a positive and what is a negative test, right? If it's a color, then tell me what the color is. With a gelatin tube, well, if it becomes liquid, liquefaction is positive. So, there's no colors in that case. The carbon energy sources just means what's being broken down, right? Is it fermenting glucose? Is it breaking down starch? Does it have catalase? If it does, then, you get bubbles. So bubbles could be a positive test too. If there's an indicator in the media, which usually it indicates a color, it tells you did something get fermented or not, right? Then just tell me such and such is the indicator in the media. An inhibitor is something in the media that prevents other bacteria from growing. Some can grow, but it inhibits other bacteria. So I'd say about half of the media contain inhibitors, and if they do, then you should tell me such and such is the inhibitor. 2 of the tests use confirmations, right? So be sure you tell me if it's a confirmation test, then what makes the confirmation test positive or negative as well. So probably the easiest way to explain this would be to use a test like the mannitol salt agar. Okay, this is the first one. So, the purpose of mannitol salt to identify Staphylococcus, they cause skin infections, right? That means, when I say that, I mean, if you have a skin infection, then this media would probably be used to help identify what's causing the skin infection. Pretty straightforward. Okay. If you had a sore throat and you want to know what bacteria is causing sore throat, you wouldn't use this media, you would use the BAP media. That tells you if you have a strep infection or not. So then, in complete sentences, you say the mannitol salt media, the inhibitor is sodium chloride, the indicator used is phenol red. It's asked, does mannitol become fermented, right? That's why the mannitol is in the media. That's why you call it mannitol salt agar. Now you don't have to tell me what the enzymes are needed for this, right? But bacteria, if they can ferment the substrate in the media, mannitol, which is basically a sugar, then they need enzymes or the genes for those enzymes. If they don't have those enzymes, then they're negative for this test, right? So the positive test for mannitol fermentation media becomes yellow. If it remains red, then it's negative for non-fermenter, means it doesn't ferment mannitol. So you put that into sentences, okay? It might not be a bad idea to mention Staph aureus turns this plate yellow. So some of this information, you might write Staph epidermidis would grow on the media and it would remain red. So Staphylococcus is the genus that can grow on this. Most other medically relevant bacteria cannot tolerate the high salt. So most bacteria will be inhibited, but Staphylococcus can grow. If it turns yellow, it grew. It's probably Staph aureus. If it grows, you can barely see the bacteria growing on this plate. There's none growing here. They're basically growing on the edge. Then it's probably Staph epidermidis, right? It's Staphylococcus, but it can't ferment mannitol. All right, now I'm going to explain these concepts, right? Each lab, I'm going to go through this process, what the mannitol salt auger is used for, what the EMB, right? So in the video you'll be watching for this lab, it'll be a repeat somewhat of this, but this is just to focus on the P&P instructions. But if we go to another one, like the starch plate is a little bit more complicated. Okay, here's the purpose of it. But basically, these are already sort of like in sentences, but you know, put it in your own words, right? Starch is in the plate, in the principle. If bacteria secrete amylase, it breaks down the starch in the plate. You should mention that. Then after the bacteria grow, you add what, a indicator, or you could call it a reagent. Most indicators are already in the medium, but this one gets added to the surface. It gets absorbed, it stains the starch, right? So the clear zone means there's no starch in that area, and that means that they secreted amylase. Where E. coli grew here, it didn't break down the starch, so there's no zone around it, like you see this yellow or orange zone around. See, the white area is the bacillus growing on the plate. So they didn't grow in one colony. They grew in this one big long. They probably took their loop and touched there and just came down. Touched here, came down, and then, right. So E. coli is growing here. It looks white too. Okay, so that's it for, right. Be sure you complete these. They're in the quiz section, but they're not quizzes. Hopefully that's not confusing. And if you're having issues with this and you want to, you know, email me your principal to make sure it's correct. I don't mind doing that, you know. So hopefully everything's going well.